Hello friends, welcome back to a new video, that is MCQs on biochemistry, and this is the part 8 of this series of videos on biochemistry. So the first question is question number 141. So the question is, the order n for a given substrate concentration in an enzyme catalyzed reaction following Michaelis-Menten kinetics is, the order is option A n equals to 1, option B n equals to 0, option C n is not defined, option D n is greater than 0 and less than 1. So the right answer, that is the right order of Michaelis-Menten kinetics, is option D, that is n greater than 0 but less than 1. So that is the first order reaction. So Michaelis-Menten is a first order reaction where n is greater than 0, less than 1. Okay, so let's move on to question number 142. So the process of removal of electrons is called as option A reduction, option B oxidation, option C induction, option D mutation, and the right one is option B, that is oxidation. So the process of removal of electrons is called as oxidation. Okay, so let's move on to 143, that is the process of addition of electrons is called as option A reduction, option B oxidation, option C mutation, option C induction, and the right one is option A, that is reduction. So the process of addition of electrons is called as reduction. Okay, so next one is 144. So which one of the following amino acids residues is specifically recognized by chymotrypsin during peptide bond cleavage? And the options are option A phenylalanine, option B leucine, option C valine, option D aspartic acid, and the right one is option A, that is phenylalanine. So which one, that is phenylalanine, is the amino acid residue which is specifically recognized by chymotrypsin during peptide bond cleavage. Okay, so let's move on to question number 145, that is an amino acid has one proton donating group in the side chain. So one amino acid which have proton donating group in their side chain, and the pKCOOH, that is pK value or pH value, the pK value of COOH, pK value of NH2, and pKr value, that is the isoelectric point value for the amino acid, is 2.19, 9.67 and 4.25 respectively. So 2.19 is for acid, that is COOH, and 9.67, that is pK value for NH2 group, and 4.25, that is pKr value, that is the zwitterionic form or isoelectric form where net charge is 0. Okay, so here the question is, which one of the following statement about the amino acid is correct? And the options are option A, that is majority of the molecules will have a net charge, that is -1 at pH 7, or option B, that is majority of the molecules will have a net charge of 0 at pH 4.25, and option C, all the molecules will have a deprotonated R group at pH 3.22, and option D, during titration with a strong base, deprotonation will start with the R group. So what is the right answer? So the right one is option A, that is majority of the molecules will have a net charge of -1 at pH 7. How? Here we can see. So here we can see this is the first point where the COOH group. So here you can see this is the whole protonated form in the low pH, that means in 2.19, in 2.19 pH the amino acid is present in its whole protonated form. So where the net charge is +1, that is here you can see in the NH3 plus group. So this is the +1 net charge, Now in next, when that is 4.25, 4.25, they deprotonated, they release their hydrogen from first COOH group. So here the net charge is 0, that is one negative group, one positive charge. So net charge is 0. Here the amino acid present in its zwitterionic form or isoelectric form. So that is, it is the pKr value. Okay, so that is the 4.2. And next in 9.7 or 9 point, what is there, this is 9.67. so in this condition another hydrogen is released, and here it is in -1 form, and here also you can see it is in net charge 0. Okay, net charge is 0, not no charge, that is net charge is 0. So here we can see it is a -1 after 4.5 pH, that is around 9.6, that is -1 charge. Okay, so here we can see the majority of the molecules will have a net charge of -1 at pH 7. So in pH 7 most of the molecule will be -1, they will release their proton, and that is the deprotonated form. Okay, so that will be the -1 charge. Okay, so this is the right answer. So let's move on to another question, that is the carbohydrate that cannot be hydrolyzed into simpler form is or are referred as option A monosaccharide, option B disaccharide, option C oligosaccharides, option D none of the above, and the right one is option A, that is monosaccharide. So the carbohydrate that cannot be hydrolyzed into simpler form is or are referred as monosaccharide. So the next one is question number 147. The condensation product of two monosaccharide unit is called option A monosaccharide, option B disaccharide, option C oligosaccharide, option D more than one is correct, and the right answer is option B, that is disaccharide. So the condensation of product of two monosaccharide unit is called disaccharide. Okay, so let's move on to question number 148. So which of the following is or are oligosaccharides, and the options are option A maltose, option B triose, option C maltotriose, option D sucrose, and the right one is option C, that is maltotriose. So which one of the following are oligosaccharide? The maltotriose is the oligosaccharide, maltose is disaccharide, triose is a monosaccharide, and sucrose is a disaccharide. So maltotriose is a oligosaccharide. Okay, so let's move on to question number 149, that is Ramachandran explained the possibility of the protein structure on the basis of option A inductive effect, option B endomeric effect, option C steric hindrance, option D all of the above, and the right one is option C, that is steric hindrance. So Ramachandran plot explained the possibility of protein structure on the basis of steric hindrance. So let's move on to question number 150, that is phosphofructokinase 1 is inhibited only by option A ATP only, option C ATP and citrate both, option B hexokinase, option D isocitrate dehydrogenase, and the right one is option C, that is ATP and citrate both. So phosphofructokinase 1 is inhibited only by ATP and citrate both. So the next one is 151, that is consider the following statements about enzymes and which are the correct. So following statements, first read this following statement and choose the correct one. So the first option is most enzymes are proteinaceous although some are RNA molecules. Next one, The specificity of an enzyme is due to its well-defined three-dimensional structure and the active site present in it. Option 3, that is, prosthetic group is loosely bound to the apoenzyme part of the enzyme. And option 4, both portions, prosthetic group and apoenzyme are enzymatically inactive when separated from each other, and the right option is here given there as option A 1, 2 and 3 are correct, option B 1, 3 and 4 are correct, option C 1, 2 and 4 are correct, option D 2, 3 and 4 are correct. So the right answer is option C, that is 1, 2 and 4 are correct. So 1, 2 and 4 we will see. One is most enzymes are proteinaceous although some are RNA molecules. This is the right one. 1, 2, that is the specificity of an enzyme is due to its well-defined three-dimensional structure and the active site present in it. It is also true. And the last one, that is both portions, that is the prosthetic group and apoenzyme, are enzymatically inactive when separated from each other. This is also true. Okay, so these are true options. So the option C is the correct one. So move on to next question, that is 152. Which one of the following is an example of an enzyme group called transferase? And the options are option A acetyl-CoA synthetase, option B alpha amylase, option C fructose bisphosphate aldolase, option D 3-phosphoglycerate kinase. And the right one is option D, that is 3-phosphoglycerate kinase. So 3-phosphoglycerate kinase is an example of an enzyme group called transferase. So move on to next one, that is 153. In alcoholic fermentation by yeast, the NADH2 produced during glycolysis is used to reduce option A acetaldehyde to ethanol, option B NADP to NADPH2, option C pyruvic acid to lactic acid, option D lactic acid to pyruvic acid. So the right one is option A, that is acetaldehyde to ethanol. So in alcoholic fermentation by yeast, the NADH2 produced during glycolysis is used to reduce acetaldehyde to ethanol. Okay, so the next one is even though most intestinal bacterial energy source is fermentation, their growth is not limited by the low efficiency of fermentation because they have option A unlimited supply of O2, limited supply of CO2, unlimited supply of nutrient, option D none of the above, and the right one is option C, that is unlimited supply of nutrients. So even though most intestinal bacteria energy source is fermentation, but their growth is not limited by the low efficiency of fermentation because they have unlimited supply of nutrient. So next one is 155, that is strychnine, a convalescent poison, is an allosteric inhibitor of the glycine receptor. So, which of the following indicates its mode of action? And options are it binds and lowers the affinity of the glycine receptor for glycine. Option B, it changes the conformation of glycine such that it recognizes other receptors. Option C, it binds at the glycine site in the receptor, acting as a partial antagonist. Option D, it binds to a transcription factor to regulate expression of glycine receptor. So the right answer is option A, that is it binds and lowers the affinity of the glycine receptor for glycine. So strychnine, a convalescent poison, is an allosteric inhibitor of the glycine receptor whose mode of action is it binds and lowers the affinity of the glycine receptor for glycine. Okay, so the next one is 156, that is, why is methanol poisoning treated with ethanol? So options are: option A, ethanol simply dilutes the concentration of methanol; option B, ethanol complexes with methanol to form a non-toxic compound; option C, ethanol binds competitively to the target protein of methanol; option D, ethanol forms a covalent inactivating complex with methanol and target proteins. So the right one is option C, that is, ethanol binds competitively to the target protein of methanol. So that's why methanol in methanol poisoning, ethanol is used as a treatment. Okay, because ethanol binds competitively to the target protein of methanol. Okay, so let's move on to next one, that is, an enzyme has two binding sites for an inhibitor molecule. Okay, so when the inhibitor binds to the first site, the dissociation constant of the inhibitor for the second site increases, so that's leading to negative cooperativity. So the Hill coefficient for such an inhibitor is option A equal to 1, option B greater than 1, option C less than 1, option D less than 0. And the right answer is option C, that is less than 1. So here we can see the Hill coefficient. What is Hill coefficient? So Hill coefficient, a measure of cooperativity in a binding process, that is a Hill coefficient of 1 indicates independent binding. A value of greater than 1 shows positive cooperativity. Binding of one ligand facilitates binding of subsequent ligand at other site on the multimeric receptor complex. Okay, so Hill coefficient is a measure of cooperativity in a binding process. Okay, so this is the cooperativity measurement. Okay, so that is the Hill effect. So here answer is the Hill coefficient for such an inhibitor. When the inhibitor binds to the first site, the dissociation constant of the inhibitor for the second site increases. In that case, the Hill coefficient is less than 1. Okay, so let's move on to another one, that is, warming a suspension of yeast increases the rate at which sucrose is broken down into glucose and fructose, because warming causes option A increases concentration of the products, option B increases density of sucrose on the exposed surfaces, option C increases frequency of collisions between molecules, or option D stabilizes the enzyme substrate complex. So the right one is option C, that is increases frequency of collisions between molecules. So warming a suspension of yeast increases the rate at which sucrose is broken down into glucose and fructose, because warming causes or increases frequency of collisions between molecules. Okay, so let's move on to question number 159, that is, uh, a particular enzyme loses its activity if just stored in normal saline. So, however, if normal saline contains 10 millimolar 2-mercaptoethanol, a strong reducing agent, which is a strong reducing agent, the enzyme retains its activity. What can you conclude about the enzyme from above? So what is the effect of 2-mercaptoethanol in proteins or enzymes activity? So options are: it has methionine residues that are necessary for activity. Option B, it has sulfhydryl groups that are necessary for activity. Or option C, it has disulfide bonds that are necessary for activity. Or option D, it has histidine residues that are necessary for activity. So the right one is option B, that is, it has sulfhydryl group that are necessary for activity. So what is the activity? So 2-mercaptoethanol and related reducing agents that like DTT are often included in enzymatic reaction to inhibit the oxidation of free sulfhydryl residues and hence maintain protein activity. So they maintain the protein activity by inhibiting the oxidation of free sulfhydryl groups which are present in enzyme or protein. Okay, so let's move on to next one, that is the last question of this video. This 160. So a homogenate of liver cells is centrifuged at 1 lakh G for 1 hour. So following this, the supernatant is separated from the pellet and the pellet is suspended in buffer medium. So assuming inclusion of substrate and cofactors, which of the following enzymatic activities can be measured in the supernatant? So the options are: option A succinate dehydrogenase, option B glyceraldehyde 3-phosphate dehydrogenase, option C glycogen synthase, option D aconitase. And the right one is option B, that is glyceraldehyde 3-phosphate dehydrogenase. So, which of the following enzymatic activities can be measured in the supernatant? So, in supernatant, we can measure the glyceraldehyde 3-phosphate dehydrogenase activity. Okay, so these are the all 20 questions of this part 8 videos on MCQs on biochemistry. Okay, so thank you for watching this video.